Good afternoon, all. I welcome you all to the second session. And today we have with us Dr. Suprasanna. Sir is the professor in Bhabha Atomic Research Center. And the topic today chosen for discussion is radiation technology in sugarcane improvement. Sir, you are audible as well as visible, and also your presentation is visible. Please start, sir.
Okay. Good afternoon to everybody. First of all, let me thank the organizers and Dr. Ratna uh, for giving me this opportunity to interact with you all. I will share some of our experiences in using radiation technologies for sugarcane improvement. To begin with, I'll just give you a brief introduction about radiations, and then induced mutations, how they can be useful. Then we will start with our work on induced mutations in sugarcane. These will have few slides where I will just show you some introductory information. Here you can see how the evolution has began from the time before 10,000 BC and the Green Revolution during 1960s. You can see how we have evolved, how science has evolved. If you see below, these were like domesticated crops, and then later on you can see on the far right side of my screen you can see Professor Neil Rutger. He's a rice geneticist and one of the first scientists to have worked on semidwarps in rice, induced semidwarps in rice. You can see how he is showing that dwarfs induced by mutations as well as the parent variety. This is just to show you, how evolution has taken place. When we look into from the prehistoric time before 10,000 BC, and then coming back to the present time, the same goals persist. The magnitude of the problems has increased over the years. So right now we have two problems of food security and how we can sustain our agriculture scenario. These two are very important for us. In order to achieve these two over the past several decades, maybe a couple of centuries also, breeders have been following four approaches mainly: selection, hybridization, recombination, mutation. Whatever crop improvement has taken place, it has been one of these. Selection, like you select allied types in the farmer's field and then pick it up as a new germplasm line and then go to the breeding. Hybridization is like you have wild species where you try to introgress a useful gene into the cultivated germplasm. Recombination and then mutation. So I will talk to you about mutation today. So basically, people feel that mutation could be the driving force behind evolution that has taken place. And without mutation is normally a change, a modification that we see. So here you can see from the earliest maize relatives that were available, teosint, and then the present day corn, which is, and then you can see even in animals, the same kind of situation has taken place. So if when I told you it's a mutation is a change, genetic change. Basically, if the repair takes place properly, then there is no change. Recovery takes place in the organism. But if the change in DNA persists, then this can be a permanent change or a temporary change. If it is a permanent change, this can result into a kind of variation, genetic variability, which we often look into the germplasm, and if it is desirable, if the mutation is favorable, then we try to use it in breeding programs. As compared to the spontaneous mutation, which is very low, the process is very slow. Induced mutations, the mutation process is accelerated by using some of the mutagens. For example, radiations can cause mutations. If you see into the spectrum, the left side ones are those ones which are normally detrimental to life. But then when you when you irradiate, when you when an organism is exposed to radiations, certain changes can take place at the DNA level, which can persist. Which, if the recovery process, if the repair process is not done, these can lead to certain changes. And these changes, if they result into favorable mutations, these can be used. Uh, these can be useful for us. So here I just showed you ionizing rays, yeah, X-rays and gamma rays. As compared to the X-rays and other ones, gamma rays have more penetrance and are more useful as compared to other mutagens. Here is a typical gamma cell irradiator. Uh, and then these are the different sources of gamma ray uh, for gamma ray exposure, starting from gamma rooms where the gamma source is kept in the central position, and then the all around uh, you can see the pots which are lined up. These are exposed as the gamma source comes out. Here you can see phytotron, gamma field, gamma greenhouse, chronic gamma irradiation facility. It can be any of these facilities. Some of these facilities are available in the country. Some of them we do not have. So whichever mutation techniques, whether it is physical or chemical, we often try to improve our cultivars if they lack a particular character. If it is deficient in a particular trait, we try to enhance biodiversity. For example, if there is not much genetic variation present in a particular commercial cultivar or a useful cultivar, we try to enhance the variability. That's how we bring in new germplasm. And increasing farmers' income, I'll try to give you few examples when we go to the further slides, how it has generated a lot of income for different countries, and it has helped the farmers in many of these countries. Mutation breeding has more relevance where genetic variability is very low in a particular crop. We need to remove a particular defect which is creating problem for cultivation of an allied variety. If we have to shorten the duration, like early maturity, tightly linked genes, if you have to break the linkage, intervention in known biochemical pathways, novelty, value of a phenotypic change, generation of variability through recombination is not possible, particularly in case of vegetatively propagated crops like banana, sugarcane, and many other ornamentals. So in these cases, mutation breeding has more relevance. This will have some of the points which I have already talked about. Way to use induced mutations is to improve one or few specific characters of a variety, an allied variety. To induce a morphological marker, it could be color, it could be on, into a promising line to make it more acceptable for registration and release. To induce male sterility or to obtain within an adopted genotype some kind of changes which will bring in more applicability. And one can use different explants, different like seeds, pollen, whole plants, buds, cuttings, or in vitro cultures. Anything can be used. When you look into overall scenario globally, there are around 3,362 mutant varieties which have been released for commercial cultivation. If you see this pie diagram, the blue one, it indicates the mutants which are developed by gamma rays, and the other portion is by all other mutagens, including physical and other ones. So you can see the extent to which gamma rays have been applied for mutation induction. Here it is actually the number that is 1702 and 1600. So this is almost like till around 15 days back, this is the number which is given by uh, there is a database which is maintained by Food and Agriculture Organizations International Atomic Energy Agency. It has mutant variety database. So they keep on uh, maintaining all the records of mutational events which have resulted into a variety which are registered with them. So these are since 1960s, whatever varieties are released, these are cataloged in the database. For example, if you see different categories of crops, maximum is cereals. You can see 45%, flowers, oil crops, legumes, and other crops. So this is, as I told you, this is FAO IAEA mutant varieties database. So Indian scenario, if you see, ornamentals occupy a major portion of it, and we had very dedicated scientists who spent their lifetime in working about ornamentals. For example, Dr. S.K. Datta, who did pioneering work in when he was in NBRI Lucknow. He did a lot of work on ornamentals, particularly chrysanthemum. So that is how we have a large number of ornamental mutant varieties. We have cereals, oilseeds, pulses, and so on. If you see by the countries wise, China is the first, Japan the second, India stands third. We have around 335, maybe few have increased in the past couple of years. So this is number of mutant varieties which are released by the country wise. And here you can see, as I told you in the previous slides, in the Indian scenario, ornamental flowers particularly, they occupy the major share, followed by other crops. I just take you for a few minutes the work that has been done at Bhabha Atomic Research Center. This was one center where significant research on radiation technologies is done, and we have mutation breeding groups in cereals, pulses, oilseeds, and then vegetatively propagated crops. So we over the past three and a half decades, we have worked on several crops, and as you can see here, we have almost 46 mutant crop varieties which are released for commercial cultivation in the country, and some of these are very popular in the country. The famous and very popular ones are Trombay groundnuts. All our varieties, the BARC varieties, the first is T. T stands for Trombay. So we have mustard, sunflower, cowpea, urad bean, mung bean. Some of these are very popular in different states. If somebody is from Jammu and Kashmir, some of our mutant groundnuts are now being grown at high altitude. And then this is almost like, I think last year we have grown, and this is for the first time that mutant groundnuts are cultivated and grown there. So this is about our scenario. I thought I'll just brief you about it. Here, a few slides ago, I was talking about economic impact. Here you can see different countries, different crops, and the mutant varieties that are there, and you can see economic impact of it. For example, if you talk about India, we have one variety which is black ground. It is TAU, Trombay, Akola, Urid, and 3 other black ground varieties. So this has contributed to, here you can see, $163 million US. So this is the economic impact. For example, in Pakistan, NIAC 78 cotton variety has transformed the cotton cultivation in the country. So likewise, wheat, Japanese pear, this is a very famous example of induced mutation breeding technology. So this is, I thought I'll just brief you about the economic impact. So coming back to today's talk, sugarcane. As you're all sugarcane breeders, sugarcane researchers, I don't need to uh, uh, add anything extra into that. Uh, I think this must have already been talked about that UP is the major producer, followed by Maharashtra and other states. Uh, this is particularly when I was reading about Vision 2050 from Sugarcane Breeding Institute, Coimbatore, which is one of the pioneering centers in the country and in the world, which has done a lot of sugarcane related breeding and research. And we are very fortunate that we have such a center which has contributed significantly to sugarcane improvement. So I was reading about one particular booklet which they have published along with ICAR. So it talks about several goals which are set for 2050, and some of them are very relevant because we need to develop new varieties which will cater to different agroclimatic conditions in the country. For example, water use efficiency or salt adaptation, or different kind of crop production technologies which have to be innovated to be introduced. So these are some of the goals which have been set by the SBI and ICAR. In line with whatever I have told you, uh, uh, on here this side you can see stress tolerant genotypes which have to be developed using different approaches: genetics, breeding, biotechnologies. Finally, the aim is to develop novel methodologies to evaluate and screen cultivars. Here, uh, some of the some of the methods that we talk in the further slides have more relevance to evaluate and screen cultivars. So many of the time, what happens is that the methodologies that are adopted, they are more with the seed propagated. So vegetatively propagated, you need to develop new kind of methods. Here you can see the different stress factors, biotic as well as abiotic. Just to give you one example of soil salinity, here you can see whatever is the black portion here that is given, which is ICR given in, Department of Agriculture, Cooperation and Farmers Welfare, Government of India data, which shows these are the most intense salinity areas in India. Here you can see Gujarat, Uttar Pradesh, Maharashtra, West Bengal and Haryana. And salinity affects to a maximum extent 25% of total sugarcane area. And here sugarcane is, this is just to give you one example of the abiotic stress factors that we have talked about. So it affects yield as well as juice quality. So when we talk about different approaches that can be used for sugarcane improvement, mutagenesis is one approach. There was pioneering work done by initial breeders and biotechnologists who were there, who were working at Sugarcane Breeding Institute in Coimbatore, Dr. Srinivasan and others. They have done pioneering work. So induced mutagenesis has a lot of application in sugarcane. One can use chemical mutagens, one can use radiation. What we have tried to use is integrate mutagenesis with in vitro culture. This is again taking back to the IAA database. We have around 17 sugarcane cultivars which have been developed globally. Here you can see India. We have almost 5 mutant varieties which have been developed. This is the entire scheme that we have developed for sugarcane. It starts with an elite cultivar which has a certain problem. We develop in vitro cultures. I'll expand on each one of these in the later slides. We optimize regeneration, which has to be very high frequency regeneration. Then one need to optimize mutagen dose. Then, if the regeneration is taking place, you have to see that you get very good mass multiplication. Then, either at the callus level or regeneration level, you need to exercise in vitro selection for biotic or abiotic. Whatever plants survive through the cycles of selection, these are hardened in the greenhouse, later in the field, and then in the field one can look for agronomic traits. This is how we have followed in the past 10 to 12 years. We have done a lot of work on sugarcane, and then we try to develop a scheme which is now being worked out in collaboration with other institutes in India. This is how we have tried to develop high frequency regeneration. Here you can see leaf rows, how one can. This is a uh, very routine and very good high frequency regeneration system, which is reported in sugarcane by many other groups. One can also get from inflorescence segments, embryogenic callus and regeneration. Here, we have also developed a direct embryogenesis system which avoids the intervention of callus. So, at the callus stage, we try to induce embryogenic callus and then irradiate it, and then develop the irradiated cultures through different cycles, which I'll talk to you in the next slide, and then regenerate and then field plant, field grow the mutants. This is EMS. One of our collaborators from USA has done EMS-based mutagenesis in sugarcane and developed very good mutants, which are now under evaluation. This is the entire scheme. Whatever I talked to you in the previous one or two slides, this is an extension of it. Like for example, as I told you, optimization of embryogenic callus induction, plant regeneration, and acclimatization, which are very important to have a high frequency regeneration system. Then this is another most important for the mutagenesis experiment. Study of radiosensitivity of embryogenic callus or in vitro culture. You need to study this, establish a particular dose, which we call it LD50, lethal dose 50. The same thing for any other selection agent like sodium chloride or PEG for drought. Then take the LD50 dose and then grow the irradiated cultures. Then after that, we normally try to characterize the selected callus. Here I have written M1V1 to M1V4 or M1V5. This is V stands for vegetative propagation cycle. M stands for mutation cycle. So normally we go to V4, V5, or if it is required to go, we need to go to further generations, further cycles of multiplication. We normally try to characterize the selected callus for whichever trait we are looking, and then again expose it to the selection agent a few times to avoid any escapes. Plant regeneration, then field evaluation, screening of mutants, and then screening for different characters, agronomic or quality characters. This is almost an extension of what I have told you. Only that here the M1V1, this is in detail, it is explained here. This is a very good example of how they have obtained. This is resistant sugarcane. This is an example from Australia, and it is through chemical mutagenesis, sodium hydroxide. So this is like for example, it is written with M1V1 generation. Again, it is written V2 to V4. So it is basically here you exercise a kind of evaluation. It can be in the field or it can be in the lab. Laboratory evaluation for these many generations, it will give you a lot of advantage because you don't need that much of field space to extend. Finally, M5 to M7, agronomic performance of the selected mutants and testing for disease resistance in a hotspot. So this is how the entire phases of mutation induction, isolation of mutants, and agronomic evaluation takes place. For a crop like sugarcane, it could be any number of years. This is our own example where we try to use sugarcane popular cultivars which are grown in Maharashtra, establish very high frequency regeneration system. And then, as I told you in the previous slides, first we try to look into uh, radio sensitivity of the tissue. That's very important. And also you need to establish a particular dose of selection agent, in this case sodium chloride. So both are very important, but because when you use a particular dose of radiation and then keep it to put it for selection, you require a proper selection medium ingredient component like sodium chloride. And then we have developed irradiated plant population. Initially, we started our work with Dr. Punjab Rao Deshmukh Krishi Vidyapeeth, Akola, Maharashtra, where we have grown our irradiated plant population in the field, and then I isolated some of the mutants. And this is in collaboration with Vasantdada Sugar Institute, Pune, where we have developed again irradiated plant population of 2 or 3 commercial cultivators like CO740, CO8032, and then developed an entire array of mutants which are now under evaluation. Here you can see 86032, the different mutants which are being grown, which are under evaluation. And this is a kind of brief idea about how particular mutants are performing better as compared to the standard check as well as the parent. For example, some of our mutants are were looking far promising as compared to the parent. Here you can see how the internode length has gone up considerably as compared to the parent. One of such mutants, when we worked in 86032, we developed a lot of irradiated plant population, and then we selected them under saline field conditions, and we isolated few mutant clones which were looking salt tolerant. These salt tolerant mutants, we have taken it to the laboratory and then tried to see if they have retained the same characters. So interestingly, these mutants performed very well at the field level, under control, as well as under saline field level, as well as under laboratory conditions. Why we have grown under laboratory condition is we wanted to, maybe I'll explain to you a little later, but this is just to give you one of the salt tolerant sugarcane mutant which we have developed and which is under evaluation right now. As I told you, these 3 institutes we have done our initial collaboration and developed a very good number of mutants. These were under BARC and then that particular institute collaboration, where we have even some of our funded projects. We have BRC has a funding agency which is called Board of Research in Nuclear Sciences, BRNS. So we fund some projects in different crops. So we have different committees. One such committee is Food Security Committee, where we try to give projects up to 35-40 lakhs, where one can apply in collaboration with BRC, and then one can work on nutrition building or basic sciences. So these 2 institutes, we had BRNS projects where we had developed a large number of mutants. As I was telling you previously, how you can increase regeneration frequency when you have a irradiated cavus. Normally, when you go to higher irradiation doses, if I quickly take you back to one of my slides, for example, here you can see This is the salt concentration, but the same thing happens if you take from the 0 gray to 100 gray. The 100 gray callus normally will not survive. From 40, 50 gray only, you don't see any. For example, here you can see 0, 20, 40 gray, 60 gray, 80 gray. So here you can see the regeneration is going down after 40 gray, because the LD50 is somewhere between 20, 30 or so. So what we have done, we have tried to improvise some of the in vitro methodologies by which we can improve regeneration even at very high irradiation doses. Here you can see even 60 gray or 80 gray, you can see some of the low frequency regeneration which is happening as compared to no or death of callus in 80 gray. So these are a couple of experiments which we have done, and where we have proved that very high frequency, very high doses also you can induce regeneration frequency. This can help in the isolation of very rare mutations if there is a possibility to get them at high doses. So when we isolated mutants, we had, as I told you, there are two options for anybody. One is to put them in the field and then put them into a variety development mode, which we are doing in collaboration with our collaborators. The second aspect is how you can study these, what kind of mutant traits, what kind of mutations have taken place, and how these traits have changed, and why the mutant is performing better. This is a kind of looking into the mechanism. So we try to use different genomics techniques, and then try to develop a kind of understanding. For example, we used RNA-seq-based identification of differentially expressed genes in one particular mutant which had very good salt tolerance. This was a methodology slide, how we have adopted it. This is that particular mutant which I told you that it was performing better under the saline field conditions. So what we have done, we try to look into the genes which are very important for the mutant to perform better under saline conditions, and to our interest, there were some chloroplastic genes which were highly expressed. So we try to look into what these genes are. So these genes for photosynthesis, stress responsive pathways, and some of them secondary metabolite pathways. So we have right now we are at this stage, we are trying to isolate these genes from the mutant, and then we are trying to functionally validate them by overexpression in any other system, trying to see whether these can be really that kind of improvement. As I told you, whatever I have talked about sugarcane, this is with the collaboration of three of our collaborators: Vasantdada Sugar Institute, Dr. Punjab Rao Vaishnav Krishi Vidyapeeth, and Indira Gandhi Krishi Vishwavidyalaya, Kavardha, Chhattisgarh. For example, Dr. Om Narayan Verma is helping us with the field evaluation. Akola, we have Dr. Vaidya, and VSI, we have Dr. Devarman, Dr. Dalvi, Nikam, Dr. Babu, and Dr. Tawar. So these are the scientists who are helping us in the field trials. So now, having explained to you one part of our radiation technology, I'll just introduce you to the second topic, because this second topic will be dealt by one of our collaborators, Dr. Sunil Dalvi. Maybe in another 1 week, he will talk to you very in detail about this particular radiation technology. This is also under our collaboration. My group has done a lot of work with Dr. Dalvi, and we have developed irradiated chitosan as a biostimulant for increasing crop productivity. We have done extensive field trials in sugarcane and other crops. For example, sugarcane, I'll just show you one slide where we have done the initial work on sugarcane at the greenhouse level. You can see the normal and the irradiated kind of and sprayed sugarcane nursery. And here you can see the farmers field under different sugarcane factories. So I'll just leave it here because this is one particular topic which Dr. Dalvi is going to cover either on 7th or 8th of December in detail. I thought I'll just introduce you because when I say radiation technologies, these are two applications where we have worked. One is induced mutations, the other is developing a kind of radiation-based polymer which can be useful for sugarcane improvement. So I thank you all. That's all I have to talk today. Thank you very much. Hello. Hello.
Thank you, sir.
Hello. Yeah. Yes.
Thanks.
I was audible
There are
throughout
three
the
questions.
lecture.
Yeah, you are audible. You are audible,
Yeah,
sir.
yeah.
You are audible.
Okay.
If there are any
Okay.
questions, please right now, Sarjay, he will answer them. Thank you.
Yeah. Otherwise, also, I'll just take you to my first slide, so you can anytime you can write to me. I will answer all your queries. You can ask even now, no problem.
Level of radiation and whether seeds or only seed is radiated.
Pardon? Can you repeat your
Level
question,
of
please?
radiation, and whether
Ah,
seeds or seeds are also radiated.
yeah, sets also one can do. The only problem is that, as I told you, the way you have to multiply, the way you have to do initial M1V1 to M1V5, if you have to do with sets, you will require a lot of area to manage it. So if you can do under tissue culture conditions, all that can be like within few months you can finish everything. So that is the advantage. It is not that you cannot do. Initial sugarcane mutagenesis was done with sets. Even now, some people are doing it. They have success. The problem is that you need to have that much of area, that much of workforce to manage it. That is my point. If you can use tissue culture, everything is like hardly 6-7 months. Maybe you can finish everything. If you have very good multiplication, you can do wonderful mutagenesis and selection work. Is it okay? Have I clarified your point?
Yes, sir.
Hello? Huh?
Yes, sir. Clarified.
Yeah, are you convinced? Yeah, you're not convinced.
Convinced, sir.
Okay, thank you very much.
Any other question, please? Any other query, please
Sir,
ask.
at tissue culture, the treatment is given to callus or after seedling development?
No. It is actually whatever irradiation or chemical mutagenesis that is done at the callus level. It is done as I showed you. I don't know if I. 1 minute, I'll just show you. My slides are not moving. My slides are not moving. So whatever I showed you in the initial sugarcane, our work, there was very good embryogenic callus. That is the stage where we try to irradiate it or subject it to mutagenesis, any mutagenesis, whether chemical or physical.
Okay, thank you, sir.
Thank you.
Any other query? Please ask.
I can do one thing. See, I'm already. I will be sharing PowerPoint. Because the size is big. I try to make it a PDF one. I'll send it to the organizers, so Dr. Ratna or NADC, along with.
Sir, we can send. We can convert it to PDF. You can send any which way. We'll keep it as PDF.
Yeah, I have already done it. I will send it to you.
Thank you, sir.
I have already converted it. But what I'm saying is that along with that, I'll send you a couple of papers on sugarcane, our work on mutations, which
Okay,
all the participants can go through, and then if they have any queries, anytime they can write to me, anytime. And I'll be very happy to help whichever way it is possible. We will help. And as I told you, I'm sorry to take a few more minutes. What I want to tell you is that BARC and Department of Atomic Energy has a funding agency, which I told you it is Board of Research in Nuclear Sciences. So BRNS will provide funding to very good proposals which are submitted, and these can be submitted anytime throughout the year. There is no particular time. So anytime one can submit. The only condition is that this particular proposal has to be prepared in collaboration with the BRC scientist. That is the condition. So one can I leave. It is an interest to everybody. So I leave it to you. If any one of you are interested in developing it, we will help you. No problem. So
any other queries, sir, from the participants? If there is any query, please ask. We have few more minutes, and then we can consider this session to be concluded. Any other question, please ask. Sir, I think there are no further questions from the
yeah, right.
On behalf of the NADCL, on behalf of SBI, and all on behalf of participants, sir, I would like to express our heartfelt gratitude and thanks.
Thank you.
For joining us, and it was such a wonderful presentation. Thank you, sir.
Thank you very much. I'll send you the PDF to you, so you can.
You can revert back on the same email, which
yes, I'll send it to you. I'll send it. Thank you very much. Thanks
to
everyone.
Let's join back tomorrow. Thank you.
Thank you so much, sir. Thank you so much.